Recording. Share the screen. Are there any questions? So let me remind you, we're going to finish chapter 4. And that's really it for today. The practice quiz must be taken today. And there will be a quiz this Friday. Quiz 1 will be this Friday. If for some reason this takes much longer than I think it will, please let me know at 3:45 that it's time to end. I really don't think this will take an hour, but we'll see how long it takes. All right. Any questions? If not, let's go ahead and get started. Is the meeting recording?
Yes.
Okay, thank you. So let's talk about some of the structures we find in eukaryotes. The cytoplasm is pretty similar. The only difference being the cytoplasm is the part of the cell inside the cell outside the nucleus. And in bacteria, it's all of the inside of the cell. So that's the only real difference in the cytoplasm. The eukaryotes have a bunch of organelles that prokaryotic cells do not have. And prokaryotes also have something that I used to say, come on, let's start. Have something that I used to say wasn't found in prokaryotes. And in the last 10 years, they have found a cytoskeleton in prokaryotic cells. But the cytoskeleton in prokaryotic cells is very simple compared to the cytoskeleton in eukaryotic cells. So in eukaryotic cells, there's proteins running throughout the cell, running throughout the cytoplasm. And we call this the cytoskeleton. It's made up of 3 types of protein filaments: microfilaments, intermediate filaments, and microtubules. And I really won't talk much about these, but it's sort of important to know that the cytoskeleton is made up of 3 types of protein filaments. And here you can see the cell, sort of. And the cytoskeleton is almost everywhere. This cell has probably pseudopods, and you can't see the cell membrane, but it's probably something like this. And this whole region is the cell with pseudopods here. And the cytoskeleton, as you can see, is almost running everywhere. In this cell, which is pretty distinct over here, it gets a little less distinct on this side. Where is the boundaries of the cell? But that's because there's more than one cell there. They might be nuclear cells, in which case they have their cells stretching out in different ways. And it looks like this region is where the nucleus is, and why the cytoskeleton is different in this region as the nucleus is there. Okay, just to show you a little bit about the cytoskeleton. Any questions about the cytoskeleton? Like I said, prokaryotic cells do have a cytoskeleton with proteins running throughout their cell, but it's nowhere near as elaborate. And this is not all of the proteins. This one here is only F-actin. This one here is, come on, what is it? Sorry. Actin filaments shown in red, microtubules in green. So this one's showing you 2 of them. Is that the nucleus in blue? I can't see that. Yeah, the nucleus is in blue. I got that right. So Eukaryotes have ribosomes, as do prokaryotes. The difference is that the ribosomes are larger in eukaryotes. They're 80S in size. They're the site of protein synthesis. They can be membrane bound or free. And in prokaryotic cells, most of the ribosomes are free ribosomes. They're just in the cytoplasm. But in eukaryotes, they can be membrane bound and they can be attached to the rough endoplasmic reticulum. There can be some also on the nuclear membrane, but it's mostly the rough endoplasmic reticulum that has some ribosomes. And that's how we distinguish the rough endoplasmic reticulum from the smooth endoplasmic reticulum. The smooth endoplasmic reticulum does not have any ribosomes attached to it. All right, I also am reminding you that eukaryotes do not have 70S ribosomes except inside their mitochondria and their chloroplasts. And we already explained that chloroplasts and mitochondria appear to be bacteria cells inside a eukaryotic cell, and that's why they have 70S ribosomes. And eukaryotic cells have organelles, and when I use that term organelle, I mean membrane-bound organelles or membrane-bound structures where the compartment of a eukaryotic cell is divided up by membrane-bound organelles. And that name is so difficult, I don't want to call them membrane-bound organelles from now. I'm just going to call them organelles, and you understand that I'm talking about membrane-bound organelles. Each of the organelles in a eukaryotic cell has a different function. They have characteristic shapes and perform specific functions for a eukaryotic cell. Their functions are involved in cellular growth, maintenance, and reproduction. For all of them that we're going to talk about, it's really maintenance. I'm trying to think of one organelle that's involved in reproduction other than that the organelles have to divide if the cell is going to divide. And I suppose you can say the endoplasmic reticulum is important to cellular growth because it's making proteins and lipids for the cell. But most of the organelles, their function is maintaining the well-being of the cell. Here we're looking at both an animal cell shown on the right and a plant cell shown on the left. And the organelles and red boxes around them are structures you should let me blow this up, I can't read it. You should know the structures surrounded with the red boxes. So if you don't know these, you should know these organelles. I'm not sure why the animal cell has a flagella, because flagella can be in some plant cells, mainly reproductive cells. And flagella in human cells is only on the sperm and in the cells of the throat, the mucous membranes of the throat. All right, any of these that you should know or ask a question about? We'll talk about the ones in red boxes in this lesson. The nucleus of a eukaryotic cell contains the DNA and the chromosomes. Its function really is to protect the DNA, as well as separate it from the rest of the cytoplasm, but mainly to protect the DNA. The endoplasmic reticulum, also called ER, its function is to help make and modify proteins, as well as make lipids. And there are smooth two types of ER. The smooth ER with no ribosomes attached, and the rough ER with attached ribosomes. And if you remember, ribosomes make proteins. And so the rough ER is involved in the making and modifying of proteins. And the smooth ER is not. It's involved in the making of lipids for the cell. The Golgi complex modifies, sorts, and packages proteins received from the ER, meaning the ER first makes proteins, mainly the rough ER, and with ribosomes. And some of the proteins could be made in free ribosomes in the cell, but the ones that are modified by the Golgi complex tend to be made by the rough ER, and then those are packaged into vesicles and shipped to the Golgi complex. And the Golgi complex modifies, sorts them, and then packages them. How it packages them is it puts them in a vesicle and then sends the vesicle to where the proteins are supposed to be. Okay, so that's the function of the Golgi complex. It's working with the rough ER and modifying proteins. Lysosomes are a special storage vesicle, and they contain digestive enzymes and all sorts of toxic substances for killing cells. And they're important in digesting things that the cell engulfs, particularly white blood cells. They will engulf bacteria, and the digestive enzymes and lysosomes will digest the bacteria that have been engulfed by the white blood cells. Now, other cells also engulf, and they have lysosomes. And for your gut cells, for example, the lysosome will help you in digesting the food that your gut cells engulf. Vacuoles, that's not really true that they're found only in plants. The central vacuole is found only in plants. The central vacuole is the largest organelle in a plant cell, and it's for storing things, which could be food, it could be nutrients, it could be waste products, and the central vacuole actually stores all of those. And it's kind of funny that they're storing their waste right next to their nutrients, but that's the way plants do things. Animal cells do have vacuoles. There's not very many vacuoles in a cell, and it's difficult to distinguish on an electron microscopic picture a vacuole from a vesicle. The basic difference is a vesicle is something that moves things from one part of the cell to another part, and a vacuole is just storing things for the cell. So sometimes the cell could put like proteins in a vesicle and then move it to a certain region in a cell and then decide to store things, in which case the vesicle now becomes a vacuole. And when we look at a vesicle and a vacuole under an electron microscopic picture, they essentially look the same. As I stated, the only difference is a vacuole is storing things and a vesicle is transporting things around. Any question about that? Okay, a mitochondrion is the site of ATP production in a eukaryotic cell. This is where cellular respiration occurs, and you could call them the cellular powerhouse because cells need ATP to get work done, and the mitochondria is where ATP is produced in eukaryotic cells. Now, I should state that there is one exception, and that is our muscle cells can also make ATP by fermentation, and that happens in the cytoplasm. So muscle cells are an exception. Normally, our cells only make ATP in the mitochondria, but muscle cells are an exception. And even muscle cells, they only use fermentation when oxygen becomes limiting, meaning when the muscle is really working hard, that's when it will use fermentation. At all other times, like right now, my muscle cells are only making ATP by using my mitochondria because I'm not working really hard. But if I were to go out running, which I almost never do now, if I were to go out running and would do that for a long period of time, because I'm not a very fast runner and I'm not very good at running anymore, there would be fermentation happening in my muscle cells. Okay, so that was just a clarification there. For most of our cells, ATP is made in the mitochondria, but muscle cells can be an exception. Chloroplasts are only found in plant cells and photosynthetic protists like the algae. This is the site of photosynthesis. And really, that's all I'm going to talk about that for chloroplasts, because we're not really talking about plants in this cell, and animal cells don't have chloroplasts. Perioxysomes contain various enzymes for oxidation of certain organic substances. Perioxysomes can destroy H2O2, and that would be a poison that our cells can make when we're engaging in aerobic respiration. So perioxysomes can oxidize certain organic substances, and cells need them, especially animal cells, to destroy certain substances like hydrogen peroxide and oxidize other molecules. All right, any question about perioxysomes? When you look at perioxysomes, they look very similar to vesicles and vacuoles. And really, under the electron microscope, unless there's a special stain, you can't tell them from vesicles or vacuoles. But they have a different function from vesicles and vacuoles. Any question about that? The centrosome is involved in cellular division. It actually is not a membrane-bound organelle. It is a bunch of protein fibers and the centrioles, and it plays a role in mitosis. It makes the mitotic spindle. And let me end the show and go back. That was actually the slide I wanted to see, to this here, and show you what I mean, if I can see it here. So here's the centrosome, and let's see if I can blow that up. It is not a membrane-bound organelle. It is a structure found in the cell. There's no membrane about it. There's usually two of them in an animal cell, and in mitosis, they make the mitotic spindle. Any question about that? The important part is the centrosome is not a membrane-bound organelle like that vesicle or this mitochondria, where it has a membrane about it. It's just a protein structure in the cell. And I typically call them cellular structures, but you could call it a non-membrane-bound organelle, which is a term I don't even like using. All right, any question about any of that? The Golgi apparatus is a special structure in the cell that sort of looks, once went too far, sort of looks like, let me just blow it up. Sort of looks like a stack of flattened pancakes. This is an actual electron microscopic image, and here we see the artist's rendition of the Golgi apparatus, which this sort of looks like a stack of pancakes, and you can call these blobs of jelly or something, okay? The Golgi apparatus modifies and sorts proteins. The proteins are made in the rough ER, and then they're packaged in a vesicle, and the vesicles, come on mouse, form. The vesicles form out of the endoplasmic reticulum, the rough ER, and then bring the protein, the Golgi apparatus, and they join the Golgi apparatus on this side. So here we have a vesicle that's currently joining the Golgi apparatus. And the Golgi apparatus modifies the proteins, meaning chemically alters them, making them the protein you want them to be. And then it packages them and then sends them elsewhere in the cell. And how it does is on the other side of the Golgi apparatus, it buds off vesicles. So there's a bud forming, and there's a bud that's already formed. And then the bud moves elsewhere to wherever the proteins should be in the cell. Okay? Any question about that? So the rough ER and the Golgi apparatus are important in making and modifying proteins and then sorting them into packages and then delivering them by a vesicle to where the protein should be. Any question about that? And so the vesicles transport other things in the cell, and vesicles coming out of the Golgi apparatus or vesicles coming out of the rough ER are transporting proteins. But a vesicle can form elsewhere in the cell and then transport something else like lipids or hormones, okay? And then that's what vesicles do. They just move things from one part of the cell to other parts of the cell. All right. I've got a brief note about lysosomes. They store things, so they're vesicles, storing things, and they're important in digesting things, meaning they have a bunch of digestive enzymes in them. Lysosomes look pretty much like vesicles. All right, any question about any of that? So the endomembrane system is a system of structures in the cell that includes the endoplasmic reticulum, the vesicles taking proteins to the Golgi apparatus, the Golgi apparatus modifying and packaging the proteins, and then vesicles taking the proteins from the Golgi apparatus to where they should be. And this all is called the endomembrane system. And it doesn't look like I have a movie on that anymore. I mean, the movie doesn't seem to be working. Let's see if that goes here. All right, I'll just walk you through. I sort of already talked about it. The proteins get made on the rough ER, and if they need to be modified, they'll be packaged in a vesicle, and then the vesicle will bud off of the rough ER, and then the vesicle will move to the Golgi apparatus, and the Golgi apparatus will fuse, the vesicle will fuse with the Golgi apparatus, delivering the proteins to this side of the Golgi apparatus. And then the proteins will be modified in the Golgi apparatus. And when they're finalized, meaning finalized, chemically modified, they will then be put into a vesicle which will bud off of this side of the Golgi apparatus, and then the proteins will be taken to where they should be in the cell. So it looks like this is making digestive enzymes, and it'll fuse with a lysosome. For a teenager, this will be making protein hormones. They'll be modified, and then the vesicle will fuse with the cell membrane, and then that will dump the hormones into the blood of the teenager, and that is why you'll get the teenager in hormonal cycle, whatever that's called. I forgot what that's called. Any questions about that? And that explains why teenagers go into their hormonal cycles. All right. I think I've already explained that. I've already talked about vacuole. It's a vesicle that is a space or cavity and functions in storing different elements for the food. It could be storing food or structural support or storing waste. While it is moving, like I said here, it says transport, we usually give that term a vesicle. And then once the vesicle stops moving, we call it a vacuole. Okay, and let's see if I can blow that up. It might not be visible, because it's got a plant cell over here. I'm going to look and see if I can see. Yes, right here. Nope, that's the nucleus. So I'm not seeing the central vacuole. Let me go back to the plant cell right here and see if there's a central vacuole. Yeah, it's right there. In the plant cell, the central vacuole is the largest organelle in the plant cell. Animal cells do not have a central vacuole. The function of the central vacuole is to store things, and it can be nutrients and waste products. And that's just the way plant cells do things, how they store things. Plant cells have a central vacuole because they don't move and they generally don't secrete waste products. They just store them in the central vacuole. Animal cells, as you know, the animal will excrete waste, and that will be urea and the undigested food. And so that's why animal cells don't have a central vacuole. All right, let's talk about the mitochondria. I've already talked a little bit about that when I was talking about the endosymbiotic theory. The mitochondria is the site of cellular respiration, where most cells make ATP synthesis, and even of animal cells that do not, that can make ATP in the muscle cells. Even in the muscle cells, we have mitochondria, and they do make ATP, and they make the majority of the ATP even in muscle cells, even for an Olympic athlete, because even an Olympic athlete, they're only using their muscles strenuously for a certain time during the day. The rest of the day, they will be using their mitochondria for making ATP. So the mitochondria is a cellular powerhouse. Here's what it looks like, shown in an actual electron microscopic image. This is two-dimensional, that's three-dimensional. the mitochondrial has two membranes, the outer membrane and the inner membrane. The inner membrane is highly folded to give lots of surface area for the mitochondria, and it's the inner mitochondrial membrane that engages in the third stage of aerobic respiration, where the electrons are transported and the electron transport chain occurs along and across the inner mitochondrial membrane. Inside the matrix is where the Krebs cycle happens in a eukaryotic cell. Glycolysis, of course, does not happen in the mitochondria. It happens in the cytoplasm. So two membranes, and the mitochondria does have DNA, which is one of these dots. And it also has 70S ribosomes in it, which are probably all of these dots we're seeing right there. Chloroplasts are similar to mitochondria in that they have two membranes. They have the outer chloroplast membrane, and then the inner membrane, which makes the structure of the granum. And the granum is highly folded. So here you're looking at the inner membrane of the granum. That would be the inner membrane of the granum in an actual electron microscopic image. And it's very hard to tell what's inside the granum and what's outside the granum. The flattened membrane stacks, the thylakoids, arrange into the granum, and this is where the plant cell engages in photosynthesis. The mitochondria also have DNA within them. It's not shown here or here. And then 70S ribosomes, which if I blow this up, we'll probably see 70S ribosomes. Yes, these little dots are the 70S ribosomes of the mitochondria. All right, any questions about the chloroplasts and the mitochondria? If not, that's it for the lesson on the organelles of eukaryotic cells. So if there's no questions, I'll end this and I'll say goodbye. Okay, well, I will see you tomorrow.